everyone. I guess we'll get started with the material for today's lecture. So what we're going to be seeing today is something I'll try to do each week, which is to get beyond the more theoretical material and biology background we're usually talking about earlier in the week, and get into some more hands-on material with how we would actually access the resources such as databases or analysis tools that you might be using in your day-to-day laboratory work. And we're going to start that by talking about some very basic resources that you're likely to be using throughout the term and hopefully beyond this in your work, related to use of sequence data or sequence databases. And so sequence, as we remember from Monday, flows from the central dogma. So we saw that there were two versions of central dogma for prokaryotes, where we have DNA, so typically circular chromosomes. The DNA contains signals that identify transcriptional units, so operons that have regions that are transcribed. And then these operons produce mRNAs that have additional signals that identify sites of polypeptide sequences, so coding sites for proteins, which then may go on to produce some other kinds of modifications. For eukaryotes, just to remind you, it's similar but a little more complicated. We have DNA sequences again. These contain signals that identify transcriptional units that would typically correspond to a single gene. And then within these genes, we would have some intron exon structure encoded in the pre-mRNA, and additional signals within the pre-mRNAs would tell the splicing machinery that the pre-mRNA needs to be spliced in a particular way to produce a mature messenger RNA. And then that, again, would have signals that would identify that a ribosome should be producing protein sequences from this. So that basically is the central dogma, and that gives rise to the three main kinds of sequence data we'll be seeing this term. So DNA sequences, RNA sequences, and protein sequences. And a lot of what we'll be doing in this class is learning to work with various kinds of data sources, and this will be the first kind of data source we learn to work with, so sequence data. And to do that, we need some way of accessing sequence data. And that's where we get into the topic of sequence databases. So before I move on, are there any questions or anything left over from the earlier lectures about central dogma, about what sequences are or where we get them? Okay, well then, moving on, we have sequence databases, and these, of course, come in different flavors. So we have some databases devoted to genomic sequence. So a genomic sequence can be anything from a single gene to an entire genome, sometimes a collection of genomes. And we'll be looking at databases where we have pages or entries within this database devoted to these different scales of genomic information. Likewise, there is mRNA. Some databases will combine the two of them, so we will often have mRNA corresponding to transcribed regions of genes, sometimes smaller pieces of mRNA. Usually this is going to be post-splicing. It's pretty hard to get sequence of pre-spliced mRNA, but often this is going to be predicted, sometimes experimentally measured. This is something we usually want to pay attention to if we're looking at an entry. And very often we're going to see many slightly different entries. Some of these may be mistakes, some may be genuine variations in how a particular gene is transcribed, but often you'll see quite a lot of these for a given gene in a given database. Likewise, there are protein sequences. Usually we keep those in separate databases than our nucleic acid sequences. And one little detail I pointed out before, just to be aware of, is you should all know that RNA contains sequences of A, C, G, and U rather than A, C, G, and T. Nonetheless, the databases almost always use A, C, G, and T for both RNA and protein. So just be aware of that when you see it. You won't generally see U's in an RNA database. Any questions about this? Right, so databases get filled in various ways. So a lot of these databases we'll see are manually generated in the sense that there are laboratories all over the world generating sequence and placing these in these databases as the sequence is generated. Some of these are sequences directly coming off of sequencing machines. So basically, the same day a sequence is gathered in the lab, someone will upload it to one of these databases, and then it's available sometimes after a short curation delay for anyone who wants to look at it. In other cases, databases are automatically generated. We'll be seeing some examples of these. So a typical example would be someone will sequence a new genome, construct an assembly of the genome, so basically a reconstruction of what an entire organism's genome might look like, and then it will go through various what are called annotation pipelines, trying to figure out where the mRNAs, the proteins, and so forth are, and then those may be deposited in other databases. So we will see some examples of entire databases that are automatically generated through this kind of analysis. And later in the term, we'll get into much more detail about how this is done. So where these predictions come from and why that's important if we want to work with them. It's also often important to pay attention to whether a database is curated or uncurated. So curated means that human experts have gone through, looked at the entries, checked that they're correct, often added other information. Sometimes there are specialized data sets we'll see where every entry has been human curated to try to produce a nice reference set of sequences we can rely on, but that takes time, it takes effort, so a lot of the data that's out there will be uncurated, just directly deposited in a database, maybe put through some automatic checks, but not really verified. And again, that's something we need to pay attention to. Often we'll have a mixture, and you have to look at the individual entries you're trying to use to figure out if you're looking at a nicely curated entry or one that may be less reliable because it was automatically generated. Any questions about that? All right, so there are a lot of different ways that sequence gets generated, a lot of different reasons people generate sequence that ends up in these databases. Not too long ago, the standard would have been that people generally put a sequence in a database because they're looking at a particular gene, and a lab that is maybe interested in that one gene has gone to the effort to sequence it, deposit it in a database, and make sure there's a nice, good quality entry for that gene. That's not done so much anymore, and much more often today, what's ending up in the database is coming from much larger sequencing projects. Potentially looking at whole genomes, maybe many genomes at once, whole transcriptomes, or more complicated kinds of specialized sets of information, but generally much larger amounts of information. And there's a trade-off there because generally having a lot more information is nice, but if you have more information, that means it's generally been less carefully checked, and you have to be careful about that. Often the newer information in the databases is less reliable than the older information because of that. And that, again, is something we'll be returning to again and again in this class, judging the reliability of our data and looking at the evidence behind particular pieces of data. So these are things you need to pay attention to when we start working with these data sets. In some cases, we're looking at curated high-throughput data. So it might be that someone has gone through this uncurated data and tried to find very reliable pieces of it, build specialized data sets of nicely curated subsets. In other cases, we might be looking at less curated data, but for something that is a minor variation on what's already in the database. So today, for example, if you work in cancer research, you can go and find databases of thousands of tumor genomic sequences. So these may be very similar to one another, but we're really interested in the differences, and so people will go to the effort to sequence thousands of genomes and deposit these in the data sets. More and more today, we're even seeing what was called environmental sequencing. Now more often you'll see the name metagenomics. These are cases where someone is sequencing many genomes at once, often without knowing in advance what they're sequencing. A very popular example of this that's been getting a lot of attention in the scientific press is what is called microbiome sequencing. So an example of microbiome sequencing would be taking samples from the guts of human beings, so looking at lots of different people, taking samples from their guts, and trying to figure out what are all of the microorganisms in those samples. And generally what you would try to do is just purify all the DNA from those samples, run it through your sequencing machines, and then computationally try to figure out what's there, which pieces go together, and so forth. So that is some of the major ways we end up with sequence in these databases. And this sequence has been accumulating at faster and faster rates over time as the genomic era has really gotten underway. So a lot of this is driven by biotechnology. And I mentioned on Monday that there have been great advances, and there continue to be great advances in the technology behind sequencing. So it was not so long ago, so maybe 10 years or a little more ago, that the Sanger sequencing technologies I mentioned in that class were the dominant way people gathered sequence. This has been largely replaced by much faster, more high throughput methods. And so we've seen pretty much an exponential growth in these databases over time. This is showing numbers of sequences and numbers of nucleotides in one particularly important database called GenBank. And for a long time, GenBank was the repository for sequence data. Any scientist anywhere in the world doing sequencing would put their sequences in GenBank, and then any other scientist who wanted access to those would be able to work with those and see what they could tell them about the specific questions they were studying. And we could see that for many years we had this exponential growth, just more and more sequences accumulating over time. You might see that this seems to be kind of tailing off in the last couple of years, and you might suspect that maybe we're hitting the end of this growth phase. It's actually the opposite. The reason this is tailing off is because the sequencing technology is producing so much data that it's no longer practical to put everything in GenBank. So there are now new repositories, such as what's called the Short Read Archive, that are trying to collect all of this data that is coming off these sequencing machines. They're not collecting everything, but quite a lot of it. And at this point, as of, I think it was 2 days ago, the Short Read Archive now has 3.67 petabytes of sequence data. So that's a lot of data. It's a challenge just to be able to work with anything like that, but this is something to be aware of. It's really dramatically changed how people can do biological research, and this is something that people are still figuring out. This data is going to keep accumulating at faster and faster rates, and we're trying to work out what we can learn from that and how to do that. And that's a lot of what we are going to be seeing over the course of this class. Any questions about any of that? Right, so what I want to do at this point is get into the hands-on portion of this. So I'm going to break from the actual slides here and hopefully be able to do this in real time, working with some of these sequence resources. So this doesn't always work. These websites can sometimes get overburdened, but I'll try to walk through what we might do in this kind of analysis. And what I'm going to start with is a resource called the NCBI gQuery resource. So I've sometimes said in this class that the single most important piece of information I'm going to give you over the entire course of the term is this URL here. So this is the central portal you would use to access the resources of the NCBI, which is the National Center for Biotechnology Information. It's one of the institutes of the U.S. National Institutes of Health, and it's probably the best place in the world for collecting the informatics resources we would need to do modern biology research. So what this is giving us access to is a search engine that lets us look through all of these different kinds of databases. And what I've done here is open up that search engine right here so we can see some of the kinds of resources present here. And I'll start by typing in a query here for a kind of gene we might be interested in called a fibroblast growth factor receptor gene. And then I'll let it search for that. So can everyone read the text on these slides here? Okay, so this is showing some examples of hits to many of the different data resources here. And there are a lot of different databases linked to this, a lot of kinds of resources, some of which we'll be seeing over the course of this term. Actually, quite a few we'll be seeing, others of which I think you guys can experiment with and figure out on your own. So we have literature resources, scientific papers related to these queries. NCBI this links to a database called PubMed. It's a great place to look for biomedical scientific papers. We have health-related resources. So if we wanted to know about mutations in fibroblast growth factor receptors that have health consequences or phenotypes, we look there. Information on genomes that contain this, chemicals that interact with fibroblast growth factor receptors, proteins that are produced by it. But for today, what I want to do is look at gene-centric entries. So look at entries that relate specifically to fibroblast growth factor receptor genes. So I'll click on this link here, gene, which will take us to hits to a database called EntreGene, which is the main repository of gene-centric pages. So every page in EntreGene is going to talk about one particular gene and one particular organism. I'm not sure how many of these pages there are in total, but there's quite a lot of them. And they're often the single best place to start looking for information about a gene of interest. So each of these lines here is telling us about one hit to that query. You can see that we've got 1879 hits in total. And we can see, for example, there's a hit to something called FGFR2. FGFR2 is the human fibroblast growth factor receptor gene. We get some basic information on where it's located in the genome, and also a variety of aliases, so other names by which this gene is known. I'll just point out that aliases are a big annoyance if you work in bioinformatics. Many of these genes have many different names that have been given to them by different people over the years, and often you need to pay attention to that because what the gene is called in one resource might not be what it's called in another resource, and then you have to worry about that if you're trying to link together different pieces of information. So that's just an aside. What I'll do is show you one of these pages, and I'll go down to this one here, FGFR1, the human fibroblast growth factor receptor 1 gene, and click on an EntreGene page related to that. All right, so this is an EntreGene page. I've zoomed it in quite a bit, so we're only seeing a tiny fraction of what's on the page, but I just want to point out a few things of interest that might be particularly useful to you, and then we'll dig into more depth of some of the actual information behind this. So these often have some nice summary information at the beginning of the entry, nice little description of what this gene does and why it's particularly important. And then if we scroll down, lots of different kinds of organized information presented in a way that hopefully makes it easy to access and use. We get information on where it's located in the genome, what other genes are around it. There are better ways of seeing that that we'll see later in the term, but you get a quick summary. We get information on different splice forms of the gene. So what we're seeing here is a kind of visual coding of the different mRNAs or transcripts produced by this. And the way to read this is that each of these green lines here would be one transcript known for this gene. And where you see these thicker bars here, those are exons. Where it's thinner here, that's intron. And maybe a little hard to see from where you're sitting, but darker green represents coding region of these genes. Lighter green would represent things outside the coding region, so untranslated regions or UTR. So each of these transcripts would have some 5' UTR at the left end, some 3' UTR at the right end. So any questions about this yet? Right, so this is giving us a quick visual code, but there's a lot of information behind this that we'll want to access a bit later. So one of the things you can note is that there is a genomic sequence here, and we can see this label NC00006.11. That is an entry to the GenBank database. So that's an identifier that would allow us to find the piece of genomic sequence, the piece of DNA on which this gene is found. And here we can see in particular, this is found on human chromosome 8, so there is an entry for human chromosome 8. One particular release, so one particular construction of what's called the reference human chromosome 8. It's a kind of the average canonical human chromosome 8 that they just declare to be the standard reference at a particular point in time. Each of these entries here also has sequences associated with it. So these tags here that we can see that start with NM, so NM_023.106.2 is an entry to an mRNA piece of information. So when we see an NM, and we also see some XMs here, that's mRNA information. So entries also into GenBank, which contains both DNA and RNA. And we also have NPs and XPs here, and NPs and XPs refer to protein sequences. So that's in a related database called GenPep. So GenPep is collecting protein entries for these different transcripts. I'll just point out that the distinction between an NP and an XP, or an NM and an XM, so the N's are curated entries. Those are ones we more or less believe the evidence behind. The X's are predicted entries. So those are ones that may be less reliable. Typically, these would be predicted in the process of assembling the genome. So computationally predicted from the DNA sequence that was assembled here. Any questions about that? Okay, so there are various other things you can see here. I'm going to skip over some of these. If you skip over a lot of what's here, you guys can refer to it later. We will sometimes be interested in the literature behind this, so the scientific literature that tells us about the functions of these. This is giving us summaries of some papers in the PubMed database that tell us about this gene. And often what's particularly useful to us are these gene rifts, which is a kind of curated literature entry where someone has gone through the scientific literature referring to these genes and has basically written these little one-sentence summaries of what important piece of information a particular paper tells us about a particular gene. So this paper here, this is a link to a paper that's telling us that a polymorphism, a variation in this gene, is associated with the rate of developing cleft palate or cleft lip in Iranian patients. So maybe that's something we are interested in studying. We'd want to look at that. And if you clicked on that, that would take us to another database on that kind of information. So as I say, we'll be seeing a lot more of these things later in the term. Often this is going to be organized in ways that will make it easier to find particular pieces of information of interest to us. So if we are interested in looking at the GenBank entries or the GenPep entries associated with this, there will be these nice summaries, each one devoted to a single transcript, a single splice form of the gene, where we can see the mRNA entry, the protein entry, and a lot of annotation telling us about this. And again, later in the term, we'll see where some of this annotation comes from and how you would use that. One other thing that is useful on these pages is that they have a lot of links to other resources. So sometimes resources at the NCBI, sometimes others, so other databases that have different kinds of information. I'll just show one example of this that's often useful. That's called the Unigene resource. That's kind of a link to other things related to particular genes. So if we click on Unigene, it will have an entry for this gene. And Unigene tends to be a good place to look for entries on homologs of a particular gene. So if we want to know about genes that are related to this gene in other organisms. So for example, we could see here that there is the human FGFR1 gene that has an entry here, but then a lot of other organisms have genes related to the human FGFR1 gene. So we can see that there is another gene that is 100% identical over 820 amino acids. So it looks like it's a somewhat shorter version of this gene, but exactly identical over the length where they overlap. And this is found in a P. troglodytes. Does anyone know what a P. troglodytes is? So that's a chimpanzee. It's maybe not surprising that a chimpanzee version of this gene would be almost identical to the human one, since those are believed to be our closest relatives. But then, as we can see, we can look at various other organisms, and as you get more distant evolutionarily, generally the sequence identity gets lower. What I'd like to do is look at one of these more distant entries. In particular, I will look at an organism we saw last time, D. melanogaster, so that's a fruit fly. It's got a version of this gene that's a little less than 60% identical. That may not sound so similar, 60%, but it's a lot more than you would expect by chance, so we can be pretty confident this is a homologous gene, a related gene that comes from some common ancestor of humans and fruit flies. And that would tell us it's probably a well-conserved gene that has some important function. So we can click on this. We can see that this gene is known as the heartless gene in flies, and I'll pick a gene summary here, which will take us to an Entrez gene page for the fly version of this gene. So I will point out that the fruit fly community tends to have very evocative names of their genes, so they call this gene heartless in fruit flies. Often the gene names in the fruit fly community are a hint about what the gene does. They're very often related to the phenotype you get if you knock out the gene. So when we see that this is a gene named heartless in fruit flies, that's probably a hint that if you knock this out in fruit flies, the heart doesn't develop, and that would tell us this is an important gene in development of the organism. Right, but we have a fruit fly entry. We have various information associated with this, very much analogous to the human entry. Generally not as much information for a non-human organism. Human entries tend to be the best annotated for hopefully obvious reasons, but we can see that we have some similar sorts of things. We can see that there are transcripts known for the fruit fly gene, and you might notice that the transcripts look a lot simpler for the fruit fly version of this gene than the human one. That's not always going to be true, but it's often the case that a fruit fly will have a simpler gene structure to it. In this case, we have just 3 transcripts, and each of those transcripts has just 2 exons. We've got 3 different exon 1s, and they actually all have the same exon 2. And we can see here that the coding region of these genes, the region that codes for protein, is actually the same in all 3 transcripts. Can anyone think of why that might be? What might be the advantage to a fly of having multiple transcripts if they all have the same protein coding region? Genes all code for the same protein. What possible use could it be to have several different transcripts?
If there's a mutation, one who's got a chance of developing properly.
Yeah, it could be that this gives the fly some resilience to mutation, that maybe one transcript is knocked out, but another can still produce genes. Very often, these might be under different regulations. There might be regulatory signals in that differing set of DNA there. So when the RNA is produced, maybe these extra signals control how long it survives or where it operates. So that may be another reason we have this. But basically, that's what we're going to assume we have here. And what I'd like to do is use these simpler gene structures rather than the human ones to get into really seeing some of the data behind these databases. So in particular, what I'd like to do is scroll down a bit to get some information about the different sequences related to these transcripts. And let's take isoform B, so that's the top one in that particular diagram we're looking at. And I'll just quickly go through some of the things we see in these different entries. So I'm going to start by clicking on the NP link. So that is the link to the GeneCentric, or the amino acid sequence database. And this is going to show us the actual information behind this. So we get some basic information that's telling us that this is 729 amino acids. The AA here is telling us this is an amino acid sequence, some information on where it comes from, and then lots of information that may be interesting to us if we're studying this, so in this case, literature information. And if we scroll down, we can get some actual structured information on what this particular gene is doing, so what the function of this gene is, or at least what information it contains. So we see something labeled source 1 to 729 here. That's telling us that this is an entry that contains 729 characters. We see protein 1 to 729. That's telling us that the entire source consists of a protein sequence, and CDS is a way of referring to the coding region within a sequence. Again, it's telling us the entire thing that comes from the coding region of the gene. And if we scroll down, we can see the actual amino acid sequence here. So sometimes you want to adjust the way these displays work. So there are a few boxes we can see at the top here that are useful for this. So we can sometimes want to change the region shown and sometimes customize the view. So what this is showing us is that this is hiding some additional annotation. I can add that in and we can see what that looks like. So there is some information on conserved sites within this protein. And we can see that this protein has a site that is given by amino acids 422 to 426, 430, 441, 443, 474, and so forth. And those are all contribute to something called the active site of the protein. So the active site would typically be the thing that is performing a catalytic function. If this were an enzyme, basically it's often what's doing the work of the protein that's telling us where that is. Often there's quite a lot of this sort of annotation associated with these entries. So one thing I will point out is that you might find these kind of hard to read, and the reason is that these aren't really designed for human beings to read. They're in a very structured format that's designed to be easy to read by a computer program. Those of you who are in the O2 recitation today, I'm asking you to try a simple problem to go through where you'd actually parse some of these things and learn a bit about these proteins by automatically running through these entries. But basically that's why these have these very structured formats that are maybe hard to read for a human, but are much less ambiguous than the Entree gene pages or the like, where we can also see this information. One other handy tip that we'll sometimes want is, often we'll be interested in cutting and pasting a sequence. We actually just want the sequence. To get that, what you want to do is go to the top here and click on, so I clicked it accidentally, but click on the thing that says FASTA. So F-A-S-T-A. FASTA just means put it in this plain text format where you've just got the sequence. You can cut and paste that and use that in any analysis. So any questions about any of this? Okay, well, if we go back then, we can then see what would be different about an mRNA entry. Go back to the Entree gene page, and now pick NM 079670.3. This would be the mRNA entry for this isoform. And what we can see is that it's structured very similarly to an amino acid entry. We have now 38 base pairs. That would be the mRNA transcript. We have a tag here that is telling us this is in fact an mRNA, and then some fairly similar kinds of information to what we had seen before. So if I scroll down here, you can see that we have a source, in this case 38 base pairs. We have a gene contained within that. The entire source is a gene. And then within that, we have a coding region. So 1134 to 3223 is the coding region within that gene. We could see the translation of the coding region, which is exactly the protein sequence we saw before. And we could see the nucleotide sequence. And again, if we wanted to cut and paste from that, we wanted a copy of that, we could scroll to the top, put this on FASTA format, and we could then cut and paste this and use it in other analyses. And I'll show you an example of that in a few minutes. Any questions at this point? Okay, so as we're stepping back further, we're seeing more and more complicated kinds of information because, of course, what's in the protein is implied by the RNA, and what's in the RNA is implied by the DNA. So we'll now go to the DNA sequence. We'll click on this GenBank entry for the genomic region, and we'll see what's in a DNA entry for this. So one of the things we can note is that the DNA entry is only showing us a little piece of it. So it's just showing us the piece of the DNA that contains this gene. So the actual entry is an entire Drosophila chromosome. So all the Drosophila chromosomes are there. This happens to be on chromosome 3R of Drosophila. But we can scroll down and see some very similar kinds of information. I guess I'll just point out it says DNA here. It's something you can check to know that you're looking at genomic information. And then we can see that just as before, this contains a source. That's the entire sequence we're looking at, 7769 nucleotides. It contains a gene. So in this case, it is zoomed in, so we're seeing just one gene within that source. And then we can see that this contains an mRNA. And what this is telling us is that the mRNA consists of the join of 3619 in this case to 3755 and 4415 to 7769. Can anyone tell me why this is in two pieces? Why do we have two subsets of the DNA joined together? What would those pieces correspond to? Like what is 3619 to 3755? Yes, that's an exon. That would be exon 1 in this particular splice form. We have another splice form up here, so exon 1 is 1 to 483, 4415 to 7769. And if we scroll down, we can also see where the coding region is in these. So each of these will actually have the same coding region, 5065 to 7254. And we could, if we keep looking, we'd see more or less the same information we got in the protein and in the mRNA sequences, and the genomic DNA contained within this region, in this case incorporating the additional nucleotides in the introns. Any questions about any of this? Right, so if we wanted to, we could put together a gene structure diagram for any of the splice forms of this gene. So, for example, we saw that if we were interested in that first isoform, the isoform A, we saw that we have a region of gene here. This is running, as we had seen, over bases 1 to 7769. We can see that within that, this particular isoform is running from 1 to 483 and 4415 to 7769. So we would know from 1 to 483 there is exon 1, 4415 to 7769 is exon 2. And by looking at the coding region, we could see where the UTRs and where the coding region are within this gene. So the coding region we can see from the CDS line is 5065 to 7254. So it's the 65 to 7254. And that would tell us that the protein is coded in here. And what would that make these two pieces here? Well, these two pieces would be exonic, this would be spliced out, but this and this together would make up the 5' UTR, and this piece here would be the 3' UTR. Any questions about that? Okay, so that basically is what's in one of these entries. And one of the things I wanted to do in this class is show you what we can actually do with this. So go through a pretty simple example of how you might use this kind of thing in your laboratory work. And what I wanted to talk about is an example of how we might conduct a very typical task in laboratory studies, and that is primer design. Very often, if you're actually working with a sequence, if you're interested in studying this kind of sequence, one of the basic tasks you need to do is have a way of copying or amplifying the sequence by PCR, and to do that you need primers. So a primer, for any of you who haven't seen this before, is something used in copying a piece of DNA by PCR, and primers will typically come in pairs. You would have one primer that anneals to the sequence in the 5' to 3' direction, and then the opposite one in the other direction. And these will set the boundaries of the region that's copied in the PCR reaction, forming this particular piece of genome as we're replicating it. So it's a very basic task that we would often want to do, and we might propose that maybe we're interested in doing that to study these isoforms of this Drosophila gene. So maybe, for example, we're interested in knowing where isoform B is expressed over the course of development of this fly. We'd like to have an assay to test whether at a particular point in time and in a particular tissue isoform B is expressed. So forming primers for this that will allow us to check for a piece of DNA that is specific to that isoform would be a standard way to do this. And we can use these kinds of resources with a primer design tool to help us do that. So the first thing we would want to do is go back here and identify a piece of sequence that would be useful for finding a primer to that gene. So let's say we look at the diagram here. If we want a piece of sequence that is unique to isoform B, so that's the top isoform, we'd like to create a test to a particular string of DNA effectively and figure out if isoform B is present based on whether that piece of DNA is present.
So you want primers that distinguish it from the other forms, so you want part that includes like the upstream,
Yeah, so ideally we'd want something from this exon 1 because that's found in that one isoform and there's no sequence corresponding to it in the other isoforms. So what we can do is go through and try to create a pair of primers to that exon 1. To do that, we're going to need sequence from exon 1. And we've already seen how to get some of that. We saw from the DNA entry, we actually know where exon 1 is. It's positions 1 to 483 in the gene. And so we could then go to the RNA entry and try to pull out those particular positions and then generate primers to this. So we will go to the RNA entry. We know we want nucleotides 1 to 483, so I will change the region here so that we are looking at, instead of begin to end, begin to 483. I will update the view here. And then we're going to want to work with the sequence, so I'll click FASTA here, which will give us the sequence. So this is exon 1 of isoform B. We're going to follow that? Okay, so what we're going to want to do then is pick primers. And primer design is one of those things that you can do manually, but it's really tedious and complicated. Usually what you're going to want to do is use an extra computer program to do it for you. The reason for this is that it's not entirely trivial to pick good primers. You can't just pick any random piece of sequence. So you need a piece of sequence, first of all, that will bind tightly enough to allow the PCR reaction to work, but not so tightly, because if it binds too tightly, it won't fall off when you heat the sample, which also will create problems with PCR. So that's something that is a little tricky to figure out. Basically, it depends more or less on the how much of it is A's and T's versus G's and C's, but that's one problem. You don't want primers that bind to each other. If they stick to each other, they won't work well. And you don't want a primer that folds back and binds on itself, because that won't work well. And those are things that we could figure out, but it's generally easier to use a computer program. And I just suggest one here. You may use others if you're working in a lab. So I'm going to bring up one called Primer 3+. It's a nice one with some, it's easy to use and has some nice features. I'm just going to show you right here, Primer 3+. And Primer 3+, we can paste in our sequence. Depending on what we were doing with this, we might choose different options here. So if we were trying to clone a piece of this, we would have to tell it what region we were cloning. We would pick that cloning was our task, and it would figure out if we have good primers that we can work with there. But the task here I'll just leave on detection, because what we're trying to do is just find a pair of primers anywhere in here that will let us clone out something from this sequence. So all we really have to do for that is leave this on detection, pick primers, and the program will do everything else for us. And what it's going to do then is pull up a pair of primers. It's going to show us where these are in that sequence. So we've got one primer annealing to this piece of that exon that we were looking at, another primer annealing to this piece, and you just want to be a little careful that these primers need to have opposite complementarity. So one of these will be in the direction of the sequence, so it will be exactly the same as what we see here. The right primer will be complementary to the piece of sequence we have. So the actual piece of RNA you would generate as your primer is complementary to the mRNA. So that basically is primer design. I'm not going into great depth about how primer design works, but just doing this to kind of illustrate how you might be using sequence resources in your laboratory work. Are there any questions about any of that? Okay, so one last thing I wanted to go through before we break today is just to show you that we're not always going to be using these NCBI resources. There are a lot of great resources there for many things that are the best thing out there, but sometimes we will want to use some others. So I did just kind of want to quickly go through one last database, and this is called the UniProt database. The URL is here, www.uniprot.org. And this is really a collection of two sequence databases called SwissProt and Tremble. So SwissProt is a curated amino acid sequence database. It tends to be a really good place to look if you want to know about your proteins, and especially if you want to know things like the difference between the precursor and the mature proteins, which generally has very good annotation on modifications, post-translation, that kind of thing. And that's linked to Tremble, which is more or less the same format of database, but is an uncurated version. So SwissProt, experts have gone through, carefully curated, and added information to the entries. Tremble is automatically generated, so amino acid sequences come to their database, and lots of automatic processes run to try to add annotation without human beings having to see this. And just to show a couple of examples of what these look like, I will type in part list here, so that live gene we're looking at. You can bring up some hits to the heartless query. You can see that there are a few versions of heartless in different organisms, but I will just bring up the Drosophila one here. We can note that there is this gold star thing here that's telling us this is a Swiss prot entry. The blue would tell us about a Tremble entry. And a Swiss prot entry, what we can see, will have a lot of different useful information here. So it's going to give us information on things like particularly important features in the sequence of this, so binding sites, active sites, and so forth, information on likely function of the gene, other things we'll be seeing a bit later in the term, information on post-translational processing. So if we want to know where this protein is glycosylated in its functional form, this is telling us about all the sites where these sugar groups have been attached to it. So it's a great resource if you want to know about proteins. Often it will have a lot more information than a GeneCentric entry, say. For those of you who are programming inclined, just like the GenBank pages, this nice graphical format of everything is basically an overlay on top of a purely text database that's designed to be much more easily human readable. We click on Format here and pick Text. You can see what the real Swiss prot entry beneath this looks like. So again, it's a nice format designed with very particular structure to it that makes it easy to write computer programs that can unambiguously find particular pieces of information important to us. So most of what was on that prior graphical page is contained on this page. It's just if you needed to search through it, you could download these Swiss prot entries. You can download the entirety of Swiss prot if you want, and write programs to do automatic searching and analysis of these entries. So any questions about that? Okay, so one last thing I want to show you is a Tremble entry, and I'll start by putting in fibroblast growth factor receptors, that same query we had before. And I'll also add in some advanced information. So often there's a lot more you can use to help you search for what you want. And I'll say that maybe we want a particular organism, and we'll say we're looking for it in mouse. So fibroblast growth factor receptor genes in mice. You can see that there are quite a few of these. Some of these have these gold stars telling us they're Swiss prot entries, but I will just pick a Tremble entry here. So here is a Tremble entry. So this is an unannotated, manually curated entry. And one of the things you might find surprising here is that there is actually quite a lot of annotation in the automatic entry. So there's a ton of information about what this gene is likely to be doing. We'll often have information about what it reacts with, various kinds of processing it undergoes. Not quite as extensive as we would see in a curated entry, but still a lot of information available to us. So as we'll be seeing later in the term, there are lots of ways we can figure out just from the sequence what this gene is likely to be doing, maybe how it's modified, maybe where there are particularly important structural regions, what other genes it might interact with, and so forth. And over the course of the later lectures, we'll see a lot of how this works. But you can actually get pretty far with automated annotation, but you need to be careful that you are looking at computational predictions, and those predictions aren't always correct. Any questions about any of this? Okay, so that's basically what I wanted to cover today. There are a few brief announcements before we break. First of all, you guys should all be going to recitations later today. The first recitation will be starting in just 10 minutes. Those of you who are in that one can follow Kelvin. He's the TA for that recitation. The next two are taught by Charlotte, so there's no respect there. And this is in Baker 140E, so maybe you can get the email. Be careful about that. And one last thing, everyone please remember Monday is Martin Luther King Day. It's a university holiday, so we don't have class. We'll just have 2 lectures Friday next week. Any last questions before we break? Okay, then I guess I'll see you.